Hello dear students, this is Praful and I welcome you all to One Page Biology. So in today's video, I'll be discussing with you all one very important question which had appeared in HSC board examination 2019. So let me tell you what exactly is the question. The question goes like, explain the connection link between glycolysis and TCA cycle. So let's try and understand this particular question, dear students. It's an important question. It was asked for 3 marks. So let us try and understand what is the link between glycolysis and the TCA cycle. Now dear students, before trying to understand the connection link between what is exactly glycolysis and what is TCA cycle, we need to understand what exactly is glycolysis. Now dear students, whenever I talk about glycolysis, try and understand the word glycolysis. Here the word glycolysis can be divided into 2 parts, that is glyco and lysis. What do we mean by glyco? Glyco means glucose and lysis means breakdown. So whenever we talk about glycolysis, essentially here we are talking about the breakdown of glucose molecule. Now dear students, we all know that glucose is present inside the cell, and where is this glucose coming from? It is naturally coming from the food we eat, dear students. So the glucose molecule which is present in the cytoplasm, dear students, it has to be broken down. And for what reason? It has to be broken down for the formation of energy. It is the process of glycolysis by which actually glucose will be broken down, and ultimately when it is broken down, the energy is released, which is nothing but in the form of ATP. Here the question arises is, where does this glycolysis take place? So the glycolysis occurs in the cytoplasm of the cell. Dear students, you have to remember the location, that is nothing but cytoplasm. So where exactly is glycolysis occurring? It is occurring inside the cytoplasm. Now let us understand what exactly is glycolysis. So whenever I say that glycolysis is a stepwise process, so that means glucose is going to get broken down in a stepwise manner. So that means it is not just a single step process. Dear students, there are going to be many steps in which glucose will be subsequently broken down, and finally what will be formed is the end product, which is nothing but pyruvate. Right? So dear students, you have to understand that what are the different types of products which are formed in this entire glycolysis pathway. As I said, it is a multi-step process. So there are going to be many intermediate products which are going to be formed. So let us understand one by one. Let me start with the very first step of glycolysis, which is nothing but glucose is broken down into a product which is called as, now glucose gets converted into a product which is called as glucose 6-phosphate. Now dear students, this is the first step of glycolysis. This step is nothing but a simple phosphorylation reaction. Phosphorylation means addition of phosphate, and this is basically done with the help of ATP. ATP gets converted into ADP after donating the phosphate, and that's why glucose gets converted into glucose 6-phosphate. And this reaction occurs in the presence of an enzyme hexokinase with the help of a cofactor magnesium ion. So please remember the name of the enzyme as well as the cofactor. Now the second step of the glycolysis is glucose 6-phosphate gets converted into its isomer, which is nothing but fructose 6-phosphate. Now here the enzyme is phosphofructokinase. Dear students, this is a simple reaction which is nothing but isomerization reaction. Now dear students, isomerization reactions are simple reactions in which ultimately the product which is formed has got the same molecular formula but a different structural formula. So here in this case, when glucose 6-phosphate is converted to fructose 6-phosphate is actually the isomer of glucose 6-phosphate, right? And that's why the name of the enzyme is phosphoglucoisomerase. Let's go on to the third reaction, the third step, that is fructose 6-phosphate now gets further phosphorylated to fructose 1,6-bisphosphate. Again, second phosphorylation reaction. Again in this reaction, 1 phosphate has been added, and can you guess who is going to donate the phosphate, dear students? Of course, again here the ATP is going to donate the phosphate, and ATP, when it donates phosphate, it gets converted into ADP. Now dear students, here the enzyme which carries out this particular reaction is nothing but phosphofructokinase, right? Now comes the fourth reaction, that is nothing but fructose 1,6-bisphosphate actually splits into 2 molecules. What are the 2 molecules? These are nothing but DHAP. DHAP stands for dihydroxyacetone phosphate, and the other is 3-PGL, that is nothing but 3-phosphoglyceraldehyde. Now dear students, again these 2 molecules are particularly isomers of each other and they are interconvertible. So what exactly happens is, as soon as DHAP is formed, it converts back into 3-PGL. So basically after this reaction, that is when fructose 1,6-bisphosphate gets broken down into 2 molecules, immediately DHAP gets converted into 3-PGL, and that's why after this reaction we are going to count 2 molecules of PGA, right? So in this reaction, what has happened is fructose 1,6-bisphosphate has broken down into two molecules. This reaction is also called as a cleavage reaction, and this reaction takes place in the presence of an enzyme aldolase. So now, dear students, we have two molecules of 3-PGL. What next? Now in the next reaction, 3-PGL gets oxidized, or you can say there is loss of hydrogen from that, and 3-PGL gets oxidized into something called as 1,3-di-PGA. What is 1,3-di-PGA? 1,3-di-PGA is 1,3-diphosphoglyceric acid, and this happens in the presence of triose phosphate dehydrogenase, as the reaction is simple oxidation reaction. Oxidation means loss of hydrogen, and that's the reason why 3-phosphoglyceraldehyde, dear students, has now got converted into 1,3-diphosphoglycerate with the loss of hydrogen. So remember, dear students, in this particular reaction, since there is loss of hydrogen, there is a cofactor, that is NAD, which gets converted into NADH plus H plus. So you have to remember the name of this particular cofactor, that is nothing but NAD gets converted into NADH plus H plus. Now what we have is 1,3-diphosphoglycerate with us. Now 1,3-diphosphoglycerate further gets dephosphorylated to something called as 3-phosphoglycerate. Now here, dear students, basically there is loss of phosphate, and whenever there is loss of phosphate, who is going to accept it? It will be accepted by ADP. So ADP gets converted into ATP. So dear students, you can see here that for the very first time here the ATP is getting formed. But you have to remember, dear students, that how many molecules are ATP going to form over here? There are going to be two ATP molecules which are going to be formed, because two molecules of 1,3-di-PGA got converted to two molecules of 3-PGA, right? So now we have two molecules of ATP which have been formed. Now in the next step, 3-PGA, that is 3-phosphoglycerate, gets converted into 2-phosphoglycerate. This particular reaction again is a isomerization reaction, and this reaction has been carried out by phosphoglyceromutase. The name of the enzyme is phosphoglyceromutase. Now in the next step, 2-phosphoglyceric acid gets converted, or we can say this is a dehydration reaction. So 2-phosphoglyceric acid gets converted into phosphoenol pyruvate, that is PP, and this is a dehydration reaction. This reaction occurs in the presence of an enzyme enolase. And finally, in the last step, that is two molecules of PP, that is phosphoenol pyruvate, will get converted into two molecules of pyruvic acid, and this is nothing but dephosphorylation reaction. Again, two molecules of phosphate will be lost, and in this process two molecules of ATP are going to be produced. So if you calculate, dear students, how many ATPs were formed, try to recollect. Two molecules of ATP were generated when 1,3-bisphosphoglycerate got converted into 3-phosphoglycerate, right? And another two molecules of ATP were generated when phosphoenol pyruvate got converted into pyruvate. So total 4 ATPs are going to be formed in this entire glycolysis reaction, right? So in case if the question comes as into how many ATP molecules are formed in the glycolysis, the answer will be 4. But if the question is what is the net gain of ATP, that means how many ATPs are ultimately formed, then the answer is going to be 2. Reason being, the net gain is 2 because initially, if you try to recollect the initial part of the glycolysis reaction, there were two ATPs which were used up, right? When the glucose was converted into glucose 6-phosphate, or fructose was 6-phosphate was converted into fructose 1,6-bisphosphate. So in that reaction, basically two molecules of ATP were used up. So net gain of ATP is going to be 2. But if the question is how many molecules of ATP are going to be formed, then the answer is 4. So the total number of ATPs in glycolysis are going to be 4. Now going ahead, dear students, now we know that the glycolysis means glucose is broken down in a stepwise manner, and what is the end product of glycolysis? It is nothing but two molecules of pyruvate. So now, dear students, what happens is, now comes the very important step, that is nothing but the link reaction. Now we can understand the link reaction very easily. So we know that now in the cytoplasm there are two molecules of pyruvate are going to be formed. Now what happens next is, these pyruvate molecules, they enter into the mitochondria, or they enter inside the mitochondria. But dear students, the problem is pyruvate molecules cannot directly enter inside the mitochondria. So there has to be some carrier molecule which can help in carrying the pyruvate molecule to the mitochondria. So here comes the very important link reaction, dear students. So in the link reaction, what happens is the pyruvate molecule gets converted into something called as acetyl coenzyme A. And this acetyl coenzyme A can only enter inside the mitochondria, right? So that's the reason why, dear students, until and unless acetyl CoA has been formed, pyruvate won't be able to enter inside the mitochondria, and there won't be further breakdown of pyruvate. That means there won't be further oxidation of pyruvate, and hence glucose won't be completely broken down. right? So that's the reason why this reaction, which is nothing but called as link reaction, is extremely important, because pyruvate gets converted into acetyl CoA, and further the acetyl CoA, once it enters inside the mitochondria, it can further undergo breakdown, which is via a cycle which is known as TCA cycle, right? How does the pyruvate get converted into acetyl coenzyme A? So dear students, we know that we have two molecules of pyruvate, and how exactly pyruvate gets converted into acetyl coenzyme A. So this is a simple oxidative decarboxylation reaction. Again, oxidative decarboxylation means here, oxidation means loss of hydrogen and decarboxylation means loss of carbon dioxide. So here from the pyruvate there is going to be loss of hydrogen and there is going to be loss of carbon dioxide. When there is loss of hydrogen, who is going to accept the hydrogen? Again, the cofactor NAD, which gets converted into NADH plus H plus, and of course the carbon dioxide is also getting released. And dear students, remember in this particular reaction, there is a coenzyme that is nothing but coenzyme A, which is added into the pyruvate, and what ultimately is formed, the product formed, is nothing but called as acetyl coenzyme A. So we have two molecules of acetyl coenzyme A which are going to be formed at the end of the link reaction. So now the two acetyl CoA molecules, dear students, are ready to enter inside the mitochondria, and once it enters inside the mitochondria, once it enters inside the inner portion of mitochondria, that is the matrix of mitochondria, there it enters into a cycle which is known as TCA cycle, and the full form of TCA cycle, as we all know, it is nothing but tricarboxylic acid cycle. This cycle is also known as Krebs cycle, very famously because it was discovered by the scientist Hans Krebs, and is also known as citric acid cycle. So dear students, once it enters into citric acid cycle, then the pyruvate will be completely oxidized, so there will be a lot of energy which will be formed in this entire TCA cycle. In this particular video, I hope you have understood the concept of what exactly is glycolysis and what is exactly link reaction. So in case if there is a question regarding the connection link between glycolysis and TCA cycle, so you have to ensure that you don't have to actually mention about what exactly is glycolysis in a detailed manner, or a TCA cycle in the detailed manner, because the question is not asking us about glycolysis and TCA cycle, it is asking us about the connection link. So in this particular question, you just have to mention about the link reaction, that is how the pyruvate is getting converted into acetyl coenzyme A, and you can just briefly explain about what is exactly glycolysis and Krebs cycle. So total in around 7 to 8 points you can write this answer. So dear students, in case of any comments or doubts, please mention in the comment section and I'll be more than willing to help you all. You can also mail me on my email ID. I will be putting the my email ID in the description box. You can also follow me on Facebook page, that is One Page Biology, and you can also follow me on my Instagram page, that is Praful Borade. This is Praful signing off. See you all in the next video with some another biology related important question. Till that time, take care. Bye bye. Thank you so much.